Good morning all. I am Shivanjan Palrai, Assistant Professor cum HOD of Swapna Devi College of Education, also a Chief Scientist of ABSR LABORATORIES Private Limited. Today I explain about the topics. The topics name: almost impossible to launching a patented pharmaceutical product in India, challenges and how to overcome challenges. First point is complex regulatory framework. India's regulatory environment is complex with multiple agencies involved in approving pharmaceutical products. Second point is patent protection issues. India has a unique patent law that can make it challenging to obtain and enforce patents for pharmaceutical products. Third point is clinical trial requirements. Conducting clinical trials in India can be time-consuming and costly with stringent regulatory requirements. Fourth point is price control mechanisms. India has price control mechanisms in place which can affect the profitability of pharmaceutical products. Fifth point is competition from generics. India has a large generic pharmaceutical industry which can make it challenging for patented products to compete. Sixth point is limited awareness of intellectual property rights. There is limited awareness of intellectual property rights among stakeholders including healthcare professionals and patients. Seventh point is counterfeiting and piracy. Counterfeiting and piracy are significant concerns in India which can affect the safety and efficacy of pharmaceutical products. Now I explain about how to overcome. First point is collaborate with local partners. Collaborating with local partners can help navigate the complex regulatory environment and reduce costs. Second point is develop a strong intellectual property strategy. Developing a strong intellectual property strategy can help protect patents and trademarks. Third point is conduct clinical trials in India. Conducting clinical trials in India can help generate data that can relevant to the local population. Fourth point is engage with stakeholders. Engaging with stakeholders including healthcare professionals and patients can help raise awareness of intellectual property rights and promote the use of patented products. Fifth point, implement anti-counterfeiting measures. Implementing anti-counterfeiting measures such as track and trace technology can help prevent counterfeiting and piracy. Sixth point is develop a pricing strategy. Developing a pricing strategy that takes into account India's price control mechanism can help ensure the profitability of patented products. Seventh point is monitor and enforce intellectual property rights. Monitoring and enforcing intellectual property rights can help prevent infringement and promote the use of patented products. Now I tell about regulatory requirements for herbal formulation. First point is mandatory human trials for any herbal formulation containing more than 2 synthetic-based additives. Human trials are mandatory to ensure safety and efficacy. Rationale. First point is safety concerns. the addition of multiple synthetic additives to herbal formulation can increase the risk of adverse reactions and interactions. Second point is efficacy uncertainty. The presence of multiple synthetic additives can also affect the efficacy of the herbal formulation, making it essential to conduct human trials to establish its therapeutic benefits. Now I explain about the regulatory framework. First point is compliance with regulatory guidelines. Manufacturers of herbal formulations with more than 2 synthetic additives must comply with regulatory guidelines such as those set by the Indian Council of Medical Research called ICMR or the Central Drug Standard Control Organization called CDSCO. Now I explain about the second point. Clinical trial registration. Human trials for such formulations must be registered with the Clinical Trial Registry of India (CTRI) or other recognized registry. Third point is ethics. Ethical committee approval. Ethics committee approval. Approval from an ethics committee is mandatory before initiating human trials for herbal formulations with multiple synthetic additives. As an example, a healer gel. Only healer gel has 4 synthetic additives. That's why for healer gel, human trial also mandatory. When human trial completed, this product directly launched by ABSR Laboratories Private Limited. Now I explain about the point issues with plagiarism software. First point is higher cost. Some higher cost plagiarism software making them inaccessible to many researchers and institutions. Second point is limited database. Despite their higher cost, the software have a limited databases which can lead to false negatives for patients. Third point is overemphasis on exact matching. Some plagiarism software primarily rely on exact matching algorithms which can miss paraphrased or reworded content. Fourth point is inability to detect idea plagiarism. Sometimes plagiarism software struggle to detect idea plagiarism where an author presents someone else's idea as their own. Fifth point is false positives. Sometimes some plagiarism software can generate false positives incorrectly identifying legitimate citations or references as plagiarism. Patent. Now I explain about the patent specific issues. First point is limited patent database. Some plagiarism software may not have access to comprehensive patent databases, leading to missed rejection of plagiarized patent content. Second point is inability to detect patent-specific plagiarism. This software may not be designed to detect plagiarism in patent-specific contexts such as claims, descriptions, or drawings. Third point is over-reliance or keyword matching. Some plagiarism software may rely too heavily on keyword matching, which can miss subtle form of plagiarism in patent documents. Now I explain about global patent coverage. First point is limited global coverage. Many plagiarism software may not have comprehensive coverage of global patents, potentially missing plagiarized content from international patent databases. Second point is inconsistent patent data. Patent data from different countries may be formatted or structured differently, making it challenging for software to accurately detect plagiarism. Now I explain the vital point. Challenges of drug discovery. First point is patent landscape. 99% of molecules are already patented, leaving only 1% available for discovery. Second point is difficulty in finding novel molecules. Identifying the remaining 1% of unpatented molecules is extremely challenging. Third point is time-consuming process. The drug discovery process typically takes 10 to 15 years or more. Fourth point, other molecule complexity. Often a newly discovered molecule is derived from existing mother molecules. As example, SA1 and SA2. Patent uncertainty. Even if a novel molecule is discovered, there is a high likelihood, 99%, that it is already patented, but the patent has not been published yet. High failure rate. The probability of a newly discovered molecule making it to market is extremely low due to various factors including efficacy, safety and regulatory hurdles. 7th point is resource intensive. Drug discovery requires significant investment of time, money and resources. 8th point, complexity of biological systems. Understanding the intricate mechanism of human biology and disease is essential for successful drug discovery. 9th point is regulatory requirements. Navigating the complex regulatory landscape and meeting stringent requirements for approval can be daunting. 10th point, need for interdisciplinary collaboration. Effective drug discovery requires collaboration among experts from various fields, including biology, chemistry, pharmacology, and medicine. These are the reference, and this is the main reference. Procedure and evaluation of Indian patent in India. The review article written by Sam Prakash Rai, who is also principal of Pranata Prasad Industrial Training Institute, and also an individual researcher, because individually these two researchers, one research about the biotechnology and nanotechnology, and the second individual research about the new formulation. This is my profile. Before I introduce me, I am Shivanjan Palra, Assistant Professor of Swapna Devi College of Education and Chief Scientist of ABSR Laboratories Private Limited.